Okay. Today we'll discuss about the promoter region. Okay, promoter region analysis. All we know that promoter is the important part of gene, and the promoter regions have some cis regulatory elements. We know there are two factors that control the gene. One is cis actin regulating factor. Another one is the trans actin regulatory factor. Cis actin regulatory factor mainly situated on the same gene and also targeted that gene, but in trans regulatory factor actually situated in another position or another gene and targeted different gene. Okay, so this is the situation. I will show you how the trans and cis acting regulating gene also work. Let's start, do some practical exercise, and also we can talk on function of the promoter. So in the last day, we have all downloaded the gene and also learned how to downloaded the promoter region of a gene if the gene is situated in the reverse strand or forward strand. Okay, so today I think one or two newly students they are joined. That's why they have no knowledge, I think, how to download the promoter region from any gene. So today we can again download the promoter region from the website. Okay. Uh, and another thing, my child is very small. Okay, he on my lap, so that's why he just talking, so making noise some. So I think you consider me. So I'm sharing my slides screen. I think my window is visible to all of you. And another thing, if you have any question, please type in message box. Okay. So today we discuss about the MYB gene, or I think most common gene, heat shock protein 70, HSP 70 gene is the most of the common gene. So we can discuss about the HSP gene and analyze the promoter. Okay, so let's start. So how we can download? Just we know that we go to the Google, type NCBI, go to the NCBI, select gene, type here HSP 70 gene in which and search it. So click on that. And in below we can found the protein. Go to the first sequence. Copy that first. Then go to the phytosome site. Then select tools, select blast, type here Triticum aestivum latest version, paste the protein sequence and click on go. Then we choose the first one and then click on the gene view report. Right click on that G, that is the gene view report. We can found the details of that protein and the gene. So we are not discussing here protein, we are bothered about the gene. So this is the genomic sequence. This is the CDS sequence, and we know that we need to download the genomic sequences. Also download the CDS sequences, and from that we can design the gene structure. But today we discuss the promoter region. So here we found that gene is the in reverse strand. Okay, so if the gene is situated in the reverse strand, so then we need to take 1500 BP extra from the downstream. So right here 1500 BP and submit. So we can see there in the database of that particular gene, there is the no promoter sequence available. Okay, when we are submit the 1500 BP, then it is not available. So we cannot find the promoter. So we take the another same gene but another gene from gene report. Here we found that another one, and it is situated on the forward region. Okay, so we can select 1500 BP and we can get the promoter region in the forward section. Any doubt, students or others, my colleagues or any PhD scholar in this section? If you have any doubts, please tell me. In the first situation, in there we identified one gene or we select one gene, but that gene have no the total sequences. That's why we just discard that gene. But similar gene also available in the another genome. Another we have another scientist who have submitted. So we can select that sequences, and we found that 1500 base pair upstream as a promoter region from here to here. So now we copy that promoter region into Word file. HSP 70. This is the promoter section. Okay. So now for promoter analysis, we need to go Google and write plant care. Database. This plant care actually this one, this plant care actually the cis actin regulatory element analysis tool or promoter analysis tool. So search for care. Here you submit your email ID, need to submit your email ID and paste the sequence. That sequence is no header. So we just copy the promoter risk sequence, submit here. Put your email ID and search. So after it takes some times, if the sequence length is too long, then it will take lots of time. If the sequence are too short, then it's take the less time. So wait, if the report is come, I'll share you. But maybe it will take 5 to 10 minutes. Okay. So for better understanding, I'll show you how to cis regulatory acting or trans regulatory acting regulatory factor, how to work. 1 minute. we know this is the gene structure. Okay, and here the 5' UTR, here the 3' UTR, and these green color are exome. But after the 5' UTR, there is one part that is called the promoter region. Within that promoter region, we can found data box and different cis regulatory element. Okay, now this is the another gene. Okay, and here also another. This is the HSP70 gene, and here you can found the this is the promoter region. Okay, of that gene. Now if this is the cis acting regulatory factor, okay, and that cis acting regulatory, why it is cis acting regulatory factor? Because it's control the gene of the same gene. Okay, but if this transcription factor is control the another gene of the same chromosome or another chromosome, okay, then so it is the cis acting regulatory factor for the this gene, but trans acting regulatory factor for the another gene. Okay, I think now you are all understand. So we are mostly we are targeting the cis actin regulatory factor. Why? Because it's have the direct effect of that gene. Now that cis actin regulatory factor actually controlled by the environmental stress or environmental situation. Now here the biotic stress, sorry, abiotic stress. Okay, so when abiotic stress is occur, so plants faces some problem. So some signal actually created through the plants. There is some abiotic stress like water stress or drought or other heavy metal toxicity like that. So that signal produces some chemical, and that chemical actually binds with that cis regulatory acting elements, and then cis regulatory acting element activate the promoter region and gene also activated. Okay, so this is the actually basic funda. And if we want what type of regulatory factor are present in that gene, then we can say that gene actually work in that situation. If we found after the analysis, if we see that this cis regulatory actin element actually abiotic stress factor related or environmental stress factor related or hormonal factor related, so we can say that this gene have the function of environmental stress or so now I think the result is coming. So we analyze in more details. 1 minute. This is the you can get such type of folder zip file. So you need to extract. After extracting, you can found this type of page. Okay, here you can see the details of the transcription factor. Okay, so these transcription factors, these are all are transcription factor. Okay, ABRE, ARE, cat box, G box, uh, gata box, GC box, LTR, okay, MIB live sequence, MIC, STRE, ST1, Tata box, TC reach, TGA, and some unknown factor also there, as there is a box S. So now, and where they are situated, you can see in the promoter region, they are situated is there. When you click here, they will show you all details. The situation position of that stand like that 444, and its length is 6. So you can add that 6 and it will get the from where to where they are situated. Okay, so all details of that transcription factor are available here. Now what we can do, we found that transcription factor. Okay, so how can we say that this gene actually controlled by the abiotic stress or biotic stress or other stress or hormonal, or it is among the transcription factors? Some transcription factors are, we know, that are situated in only promoter region, so we exclude that section. Like cat box, it is not a transcription factor. Okay, G box, G box is not a transcription factor. GC motif is not transcription factor. Gata is it not a transcription factor. Okay, then Tata box, this is not a transcription factor. These are the promoter region section. Beside this, there is lots of transcription factor like ABRE. Now, what is the function of ABRE? You can copy and search in Google. So, a major transcription activator module that express gene during the ABA signaling. ABA means abscisic acid. Okay, and another thing, that's transcription factor and plant response to drought stress. So ABA, we know that the ABA actually, when the abiotic stress are coming, then the ABA actually activated signal pathway. ABA signaling pathway actually activated. Now you can hear this is a member of transmit that stress signal. So we know that ABA actually the abiotic stress related transcription factor. Okay. So here how many ABA? ABRE 3. So you need to note down here the transcription factor ABRE number of 3. Okay. Then ARE. Similar way you need to find ARE. I have already prepared a database, okay, but that database for the another anthocyanin gene, this is not HBSP gene. So you can prepare your own database in Excel. You write the transcription factor name and what type of transcription factor, that is the stress related or hormonal related. So ARE, you can found environmental stress, response to environmental stress. So this ARE actually environmental stress. ARE found to it is abiotic stress. Then LTR. How many LTR is there? Only one. So LTR, I think it is abiotic stress related. It is also environmental stress response. So similarly, LTR 1 environment. Okay. So then MIB and MYC. So first we search for MIB. MIB also a gene and MIB also transcription factor. Okay. Mainly for MIB work for biotic stress or stress response. Okay, in plant. So MYB, how many? One MIB. Another one is MIB like sequence, so similar thing, and then MYC. So MIB 1, and it is also stress related. Then MYC, seed production, root elongation, leaf sensation, and stamen development. So MIC totally abiotic stress related. MIC to abiotic stress related. Now, now we analyze this. Okay, there is lots of another factor we are not discussing. So now you can see there is the 2, 3, 5, 1, 6, 1, 7, and another 9. Among the 9 factors, transcription factors, these regulatory factors, we found all are related to stress. Okay, so there may be the environmental stress or abiotic stress or biotic stress, but they are related to stress. So this gene, we have targeted HSP70. So heat shock protein, this gene actually stress related gene. Okay, and it is confirmed their promoter analysis. Now they can activate it through any one of the transcription factor can activate it, that gene. That's why one gene have the multiple function. If there is the environmental stress like abiotic stress, then HSP70 also be activated. If there is biotic stress, maybe it will be activated. Okay, so this way we can analyze total gene structure and their promoter structure. Okay, and when you so you can crop it from that page and paste in the side of gene structure. Okay, and when you are making the gene structure, then put it extra 1500 pp in the 5' UTR and show this is that you can mark it with the circle or another thing, and then you can show this is the promoter region, and within that promoter you can crop this figure like this way and paste here and give some arrow or mark that this promoter region have the this transcription factor, and most of the transcription factor are stress related. So this gene actually activated when the stress are coming. Okay, so students, any questions, any doubts?
Sir, uh, sir, I have one question.
Yeah, tell me.
Sir, uh, can we do this for a plant cell or plant chlorophyll content or any other gene, chlorophyll gene or any kind of other gene also?
You
We
are
can,
talking about the animal cell.
no, plant cell only. But can we do this the promoter region? We can do for any other gene or but all other gene or all other proteins that is transcribed.
Yes. If the sequence are available,
Okay.
okay.
Okay, sir.
First, when we are starting, you can see that one HSP gene that
Yes.
have no the promoter region sequence available, then we
Yes.
are targeted the second gene. Okay, so
Okay.
if your sequence are available, okay, promoter
Okay.
sequence are available, then you can do promoter analysis. There is no limitation, okay,
Okay.
because the transcription factor sequence are conserved. Okay, it's not
Yes.
the changes to gene. In case of
Okay.
plant, in case of animal, then there is a lots of another software, but
Okay.
this software work only the plants, and there is no boundary like that. This crop, not. If the
Okay.
your sequence is available, you can put your promoter region. And sometimes what's happen, you if you put that gene sequence, they also give you some promoter transcription factor, but that transcription factor actually not working within that gene. Transcription factor not working, that's why we need to analyze the promoter region. What's happened, in case of 1%, only 1%, you can found there is no promoter region within the 1500 base pair. Then you need to take another 3000. Okay, first you take then another 3000 base pair.
Okay.
If you have not found, then you take up to 10,000 base pair. But
Okay,
after
sir.
that 10,000 base pair, you can sure that gene actually not cis acting regulatory element. Okay, that genes have the trans acting regulatory. That's mean the transcription factors actually in the situated in another gene. Okay, so in
Okay, sir.
case of 1%, you only take the 10,000 up to 10,000, but 90, in case of 99%, it will be found within the 1500 base pair of the 5 prime. So if gene are situated in forward, then you take upstream. If gene are situated in reverse, then you take it from the downstream. Okay, and
Yes.
another thing,
We have
some
to choose. Sorry.
uh,
We
okay,
have to
okay.
choose the sequence arbitrary. And that means we have to select one after another if we don't found the upstream sequences or downstream.
If you not found the sequence, there is the no upstream in and downstream, then you choose the second gene.
Okay.
There, when you search in phytosome, there is lots of gene are present. Okay,
Yes.
so
Yes.
then you discard first one and choose the second one. Okay,
Okay, sir. Okay. Thank you, sir.
okay. And another thing, sometimes we say that some genes or some plants actually not sequenced already, and there is no database in the genome sequence. So in NCBI you can found some raw database. Okay, that is not published, but in raw database, for that you need to just go to the NCBI homepage. Here I am typing the gene. You can search like genome. Okay, and write the unknown crop. If the genome is raw genome are available, then you can found. Okay, and from that raw genome, you need to first identify the gene. If the transcriptome are available in the genome, then you can find the protein, that transcriptome, and then you analyze the promoter region. But it's quite difficult. It's need for more, I think, exercise. And if when you are working with regularly with this software, you can know better when even from me. Okay, so presently I am working on potato, taro, wheat, maize. So those are fully sequenced. That's why I'm not facing problem. But when I'm working with taro, I have facing some problem. So I am using some different bypass pathway. Okay, so you can, if you are working with the most common crop, then I think you are not facing problem. If you are facing problem of even after the complete their training, you can contact me. I will help you.
Okay, sir. Thank you, sir.
Or any,
Sir, I have one query.
yes, tell me.
Hello, sir.
Yes,
Sir, in
yes,
case
tell
of
me.
microorganisms, which software? Yes, okay. So, sir, in
Microorganism.
case of
Okay.
microorganisms, which software? Yes, sir.
Yes, yes, yes.
Which
In
software I
case
should
of
use?
microorganism, yes, yes, yes. In case of microorganism, what type of microorganism? Eukaryotic or prokaryotic?
Sir, prokaryotic.
Yeah. So that's why you know that in prokaryotic there is no intron part. Okay. So whatever in the
Yes,
gene
yes, sir.
sequence, actually that one actually transcribed. Okay. So in case of prokaryotic, you can found lots of software. Okay. In Google, you just type the prokaryotic analysis. At this moment, I am not working with the actually prokaryotic. Okay, so at this moment, I not recall any software, but I will give you in the our group what software you can use. But I don't know the promoter analysis of that prokaryotic genes. Okay, because before that I have never
Sir.
worked on that, but I know some software also available, and they are found the promoter region, and it's very easy. Okay, it's very easy. I will give you the link or name of that software. Okay.
Okay, sir. Thank you, sir.
Okay. So I think others have no questions. They are understood everything. So now this is the one of the part is actually gene. After that, we downloaded the gene sequence or we have sequenced the gene sequences. Then we do the diversity analysis. Okay, and among the similarity among the different gene sequences or any other, if you are like if you are working with prokaryotics, then you just sequence its region of different bacteria. Then you want to do phylogenetic tree analysis. Okay, so that phylogenetic tree you can analyze through the mega software. All among all of you, you know some are quite in the sorry, about the mega software, but today we are not starting the mega software. In the next class we'll started mega software, but today we are talking about another different interesting thing, that is the qPCR. Okay, or real time PCR. We heard the term about the qPCR or real time PCR. So today we are discussing the how to analyze the qPCR data. Okay, and how we can design qPCR experiment. First of thing, if you want to analyze qPCR data, because why we want to analyze the qPCR data? Because most of the software and already installed in the qPCR machine, they are not given the adequate results, and they have some problem, and they are analyzed such way these that results are not measurable. That's why we need to analyze yourself. Okay, so today we are discussing about how to analyze or how to design the qPCR experiment. Then after the talking about the qPCR, if we are getting time, then we are talking about the how to you analyze the transcription analysis, like the transcription. We know all are sorry, transcription, transcriptome analysis. We all are heard the term transcriptome. They are doing transcriptome uh, so in rice, in wheat, or lots of paper you can see, and there is lots of figure like that. But there is the one of the important part, differential expression of the gene, and how we can analyze that differential of gene expression in Excel. Today we will run after the qPCR. So let's start. What about the qPCR? How we can design the qPCR? So the basic funda of the qPCR is very simple. Normally it's like a PCR. First you need to analyze like this way. When you are doing qPCR, you have one target gene and also you have one control gene. If in case of one experiment like that, one plant, you chooses one plant that is the resistant plant for disease. Suppose spot blotch disease in wheat. Okay, so that disease is caused by the bipolar sorokina, and then you treated bipolar sorokina one wheat plant that is the resistant plant. So you treated or you inoculated that plant with the bipolar sorokina inoculum in your lab, and then the plant after the 6 hours, after the 12 hours, or 24 hours, 48 hours, different types of RNA or different types of protein actually expressed in that plant in compared to control. So our experiment situation is one control plant that we have not inoculated with bipolar sorokina, another plant that is treated plant with inoculated bipolar sorokina, and we are taking RNA or extracted RNA from the leaf at 24 hours. Okay, for what some genes we are selected some genes like MYB genes. Okay, so we want to check the expression level of MYB gene in the resistant plant compared to our control plant. So this is the situation. We can do real-time PCR. In sometimes we can do the quantify like virus inoculum or like that. But now today we are discussing about the gene expression in the qPCR. So this is the situation in qPCR. One resistant plant, another one is control, okay. Control means it is inoculated, is non-inoculated. Non-inoculated, and it is inoculated with bipolar sorokina. Okay, and we our target gene is the MYB genes. Okay, now how can we design real-time PCR in laboratory? Why I am discussing? Because this is not a part of training actually module. We are only targeted the analytical part, but if you are know how do the design, how you are designing the qPCR experiment, then you cannot do the analysis. That's why I'm discussing this part. Okay, now we all know that in qPCR plate there is a 96 well. Okay, so if you are not, then qPCR plate. So this is the qPCR plate. Okay, and this qPCR plate you can found 8 row A, B, C, D, E, F, G, H, 8 row and 12 column. Okay, so we are checking, or we are analyze the expression of MYB gene in resistant plant compared to control plant. So then we need to take, we have extracted RNA from the resistant plant and also control plant. Then we need to convert that mRNA into cDNA. Okay, and that cDNA we can check. Now, if particular MYB related mRNA is expressed in the resistant plant, then when we convert it in the cDNA, we found more number of cDNA MIB related than the control plant, and that will be amplified in the qPCR, and that will be the expressed by fluorescent like cyber green, like that, and that actually encoded in Excel or CSV file as a CT value. Okay, so from that qPCR we can found some CT value of the experimental data. Okay, so you have the resistant plant and you have the control plant. Now, when we started the experiment, there is, you need to take one reference gene. Okay, that reference genes may be the 18S gene, may be that Rubisco gene, or any other abundant gene that is the common in plant. Okay, and that is the abundantly expressed. So we can take, you can take tubulin gene, you can take actin gene. These are the abundant protein. So you need one of the reference gene. Reference gene. So here we can take actin as a reference. So actin gene is the reference. Okay. Now MYB gene, actin gene as a reference. Now you need to take another factor, that is the negative control, NC, negative control. That negative control without cDNA. Okay. So for one expression, only if for you have you have extracted RNA and converted cDNA from the resistant plant and also from the control plant. But in real-time PCR, you need to at least 3 times replication of same RNA. Okay, so for RP, you have the target gene 1 R1, R2, and R3. Three replication. Similarly, from control, you need to take 3 replication. Same, you need to the reference gene, that is the actin gene. It will be the 3 replication, and also the negative control, it will be the in 3 replication. So for checking one gene control to compare the resistant plant, at least minimum you put how many PCR? 1, 2, 3, and that is the 12. So for the experiment of the real-time PCR, you need to put 12 PCR or amplification for express analysis. Now, you know that for doing the PCR, you need the DNA, tag polymerase, DNTPs like that. But in case of real time, we got one master mix, that's everything including the cyber green, and we need to add water and just the CDN. Okay, and most of the cases the reaction will be happen in the 5 microliter or 10 microliter. Okay, so first we choose 2 column, 2 row, A and B row. Okay, so A and B row, for A we put all the control related PCR product, PCR mixture, and in B all the resistant plant related experiment. So now first, when we are like this way, R1, R2, R3 for the resistant. So for resistant, we just type here T. You can better understand. So this is the targeted MYB gene expression here. Okay, we are give the primer of MYB genes and mixture, master mix, DNTPs, or whatever that we are keeping there. And another is this is the control. So here we are also give the MIB related primer and master mix and whatever required. So these 3 actually main experiment. So this is the control. Okay. So A1, A2, A3 are the control, and B1, B2, B3 are the treated MYB gene. Okay. Now, what we have required? We required the actin, that is the abundant gene expression. For actin, AC, AC, and AC, actin and C for control. So here we are situated the flat here. Similarly, actin for treated. Actin for treated. Actin for treated. Okay. Now negative control. In negative control, there is no cDNA. So okay, for negative control, you it we are not included the data of the negative control in the qPCR expression analysis. Why? If you are found any expression in negative control, that means your experiment is wrong, because in negative control there is no cDNA. After without cDNA you found some expression, that means you are using that DAPC treated water, or your PCR tube, or like the any instrument, whatever you have used, those are pipette, micro pipette, those are contaminated. Or when you are preparing the PCR master mix like that, then sometimes we are talking without wearing any masks, so some uh, microorganism also from our mouth may be contaminated your PCR samples. So that's why we always put some negative control. If we are in negative control, we are found anything, that's mean your experiment is wrong. And if you are not getting in negative control any data, that's mean your experiment is fine. But all the expression amplification. So after the complete the amplification, you can found or you can see such type of amplification plot. And this amplification plot actually have one of the threshold level. If you are found NTC negative control above the threshold level, then your experiment is wrong. If you found the negative expression below the threshold level, then your experiment is fine. And for the each column, that is the for control, and for resistant you are found each color. For control you found one color, and for resistant plant you can found another color. So this is the actually how you can design the qPCR in lab, and then we need to analyze. Now we are talking, you found in CSV file some CT value. Okay, so we are now discussing about the CT value, and then we are analyze the experimental data. So first we have the control. Second one, we have the actin gene or reference. Then MYB gene or target gene. So this is the sample number 1, sample 1. Sample 3 or sample 5. Then resistant plant. Resistant plant. Similarly, okay. So here we are designed for the one sample, only one sample. So for one sample, you need the 12 reaction. Then for if you do the highly accurate analysis, so at least you need the 3 or 5 sample from the same plant. And so you multiply with the 12. If you have the 5 sample from control, 5 sample from treatment, then you need to do the reaction 10 into 12, that is 120 reaction. You can take 3 also. Okay. So this is the actual your data set. Now we are putting here the, you can get some CT value. So I am putting here randomly some CT value. I'm just preparing this data as a model data, not it is a real data. So you can found this type of CT value of your experiment. Okay. So you are want to analyze whether the MIB gene is highly expressed in compared to control or not. Okay. So let's do 1 minute, please. . If you have any question till now, you can ask.
Sir, from hello, sir.
Yes.
Sir, from where we can get the CT value?
When you are doing the PCR in qPCR, in
Okay,
CSV format, you can get all the CT value.
okay, sir.
Any others have any doubt? How many people are doing PCR, qPCR?
Sir, I'm doing.
You are. And Ashok, you are doing. And any other? I think no, any other are interested. So now we are doing the analysis. So first you need to, when you are getting CT value like that, okay, like this, then we need to analyze first mean of all CT value in target gene and also the reference gene. So this is the mean of the actin gene in control, and this is our target gene. Okay, so this is the main of the target gene. Sorry. Yeah. Yeah. Okay. Now we need to control. We need to mean of the control gene. So here the control reference gene is that. So we need to mean of that control reference gene. So average so 21.5. 21.27 is the mean of the control reference gene. So this is very important for analysis. First, now we first need to the analyze delta CT. So this is the delta CT. The formula is the control target gene minus CT value of the target gene minus control gene. So target gene is equal sign, target gene is that minus 21.27. Here you cannot choose this box because that value is constant for all the set. So you are found the CT value. CT value of the all, that is the control. This section is CT value of the control. This section of the resistant plant. Now, after the analyze this CT value, we need to the calculate delta CT value. So that delta CT value we need to the delta set for calculate the delta CT value. We need to the CT value of the average CT value of the control and minus the average CT value of the reference gene. That is the reference gene and that is the control. So average CT value of the average delta CT value of the control and average CT value of the target gene. So first we average the this delta CT. So now calculate double delta CT value like this way, equal sign and targeted is 1.70. Okay, so this is the double delta CT. Now the fold change of the gene expression. So the formula is 2 to the power minus double delta CT. Okay, so here this value. Enter. So this is the fold change. This is the fold change. And now you need to average this fold change for control. For control, average the fold change. And for resistant plant, average the fold change. Okay. Now this is the mean. You need the standard deviation and standard error. So standard deviation, you know that. Okay. And for standard deviation, standard error, standard deviation divided by square root of number of sample, 5 number of sample. So divided by 5. So this is the standard error. Now we can design a graph. So we select these two. Here you write fold change. Okay, now you need to put error, mode option, custom, specify. This is the error. Change the error color into red and change the rough color into solid. Change the font. So this is your expression analysis. From that huge data, you can found this type of graph. Okay, and you can see in control that gene actually express up to 2.26, but in resistant plant this gene express 3.49 fold. Okay, this is final your expression data. Now here one of the questions, whether this expression actually significantly varies or not. So these two expression are significantly differ or not. So now for that we need to do the t-test. So we can do the t-test in 1 minute. Okay, so for t-test, we can select this row, control versus the resistant plant. For t-test, you go for t-test. You can found here is the t-test. Okay, so first array is control, comma, second array is resistant plant, comma, there is tool, tails. Tails is one tail distribution or two tail distribution. So here the plant of that plant actually the same genotype. Okay, so same genotype, only the say differentiation is controlled, is treated resistant plant is treated with the inoculum and control plant is not treated, but the genotype is same. So that's why we are choosing one tail. Then the paired, one pair or two pair. Here the two pair and data set is equal. 5 in each section, 5 sample are available. So that is the two equal, two sample equal variance. So type 2, close the bracket and enter. And you can found that t value actually 0.29. So the value is the greater than of the 0.05. That's why this experiment, the data is not significantly varied. So control compared to control and resistant plant, the fold changes is not significantly varies. Okay, that's why you can write here NS in yes, and here you just write NS like that. If you are found that they are significantly varies in 5%, then you put here one asterisk sign. If you found that the significant in 0.01%, then you put here the two asterisk sign. But in our case, it is a non-significant. So this is the all about the qPCR designing and qPCR data analysis. Okay, I think it is quite tough in compared to other analysis. But if you have any queries regarding this, please ask. Or if you have any doubt regarding the analysis, I can do again. Please ask. It is one of the important topic in in field of molecular biology. I heard most of the student in field of agriculture and some are from the microbiology or biochemistry.
Sir, can you briefly explain the steps again?
Step again. Okay. Actually, nothing. The formula, if you remember the formula, I write here the formula. Okay. Then you can understood better. For delta CT, you have the CT value. You can get CT value from the CSV file. Okay. From in CSV format from the your qPCR. Okay. You can get this CT value. That means you can get this value. not mean. Okay, this value and this value you can get from your real-time PCR. Now you need to, from the CT value, you need to analyze delta CT. First, you need to analyze delta CT. The formula of the delta CT is average CT value of reference gene in control. Average CT value of target genes in control and treatment. This is the formula. First, delta CT's formula is CT value of the target gene in control or treatment, okay, minus average CT value of the reference gene in control. So see here, this is the CT value of control of acting gene. That's mean reference genes. So first we are do the average of that control CT value of the acting gene. Okay. And now we just minus of that individual CT value of the target gene. This is the CT value of the target gene. We have minus that 21.27 from the control and also from the treatment. Okay. This is the first CT value. Now the second one is the delta CT. For delta CT value, you need to take the average for double delta. So delta value of the target gene minus average delta CT value of the control. So here we are doing the average delta CT. This is the average delta CT of control, and then we just minus of that average delta CT value from the control and also from the reference. Okay, then we found the double delta CT. Now the fold change formula is FC equal to 2 to the power minus 2 to the power minus delta double delta CT. This is the FC value formula. And here we are just putting that 2 to the power minus this double delta CT. This is the actually formula. Okay. This is the actual formula. Now, Ankon Das, have you any doubt?
Yes, sir, understood.
Okay. Any others?
Hello, sir.
Yes. Ashok.
Sir, for t-test, we will take
Yeah.
control FC and rest implant FC and another two words, sir.
Another?
I have a little doubt
Okay,
in
okay.
t-test, sir. T-test.
For t-test, why you are for t-test, we are testing whatever the fold change are found in control, okay, and found in the resistant plant, they are significantly varies or not, okay. So in Excel, there is the inbuilt formula for t-test. So you write that equal sign and t-test. You can found t-test from that. Here you can found t-test. Okay. So array 1, you can see here first array 1. So which samples or which data you need to your first one. You can put control or treatment data. That is not matter ki you first put the control and then you put the resistant, not like that. You can first put the resistant plant and also put the control. So there is no boundaries. So we select that first one is that fold changes of the control data. Okay. Then comma. Then arrow 2 is the resistant plant. Okay. Then comma. Then they also said that tails. How many tails is there? So I am talking about there is the one tails. Why? The genotype is same. Control and treatment, but genotype same. So if the experiment there is the no two separate entity, then it will the one tail. If the entity is separate, if you are checking the expression data in susceptible plant and resistant plant. So susceptible plant is different genotype, resistant plant is different genotype. That's why there is the tail will be 2. But here we are using same genotype, control and treated. That's why the tail is the 1. When you put the 1, give the comma, then they ask what type of the data. That is the paired data, or 2 sample equal variance, or 2 sample unequal variance. So you can here give the paired data. Before that, I am putting the 2 sample equal variance. No, so I'm sorry. You can put the paired data. Okay, so you type 1. Okay, then bracket closed. Why 2 paired data? Because here the 5 sample, here the 5 sample. So each sample will pair with the another. That's why it will be the paired data, and you can found that value is the quite difference before that, but it is the 0.06. Okay, so if the value is 0.06, that is also less than greater than 0.05. That means in 5% significant level, it also the greater than. If the value is below the 0.05%, then we can say it is significantly varies. But here it is not significantly varies. So these two data are non-significantly varies. Okay.
Okay, sir.
Any doubt, Ashok? You do in
No, sir.
Excel yourself. You
Okay,
just
sir.
put
Okay.
some sample data in column 1 and another sample data in column 2, and then you can found t-test. In when you type the equal and type t, you can see the t-test. You just select and do this way. Okay.
Okay, sir. Okay.
Any other students have any questions?
Sir, I have one doubt, not from qPCR, but from yesterday's part.
Yesterday. Okay.
Means we have one primer, means we have not designed that primer, but we want to locate its position in the genomic sequence.
You are asking about where your primer is situated in where in
In from
your genome sequences, like
Mhm.
that.
Yes, sir. Yes, sir.
Okay, for that you need to use the phytosome. This is not our part of our study, but I'll show you. You need to go the phytosome sites. First one, if you are designing a primer yourself, then you can know where your primer or where your forward sequence or reverse sequence is situated. If you are picking primer from the another reference, then you need to search that primers where that primers are situated in your genome. But most of the cases, your genome, this result will not, you are not getting successfully, because most of the primer are sequence based. Means if you have the 10 wheat genotype and we have developed primer from one genotype which I have sequenced, and that data is not available in the website. I have sequenced and then I have designed. So then most of the cases, then we are not getting the primer in the website. Okay, but if I have designed the primer in conserved region, then you can getting that primer only. Okay, so now suppose we have designed primer from this sequence so this is the forward primer, and I have designed another primer. This is the reverse primer, but we need to change its reverse complementary. So this way you can design your primer. This is the RP reverse primer, and this is the forward primer, whatever you have. Okay, so this way you can design from primer yourself. Now you know where your primer is situated. But if you want to search by the genome site, you can take any one of the primer. If you are taking the reverse primer, then you need to convert its reverse complementary sequence. If you are taking the forward primer, then you just copy that forward primer, go to the phytosome, and then tool is blast. Then if you are know your which plant you are searching, then you are choosing latest version, paste here your sequence and go. So you can found this type of page. Now you click here. So you can see this is your gene, okay, and this is your submitted primer. Zoom out it. This is your primer sequence. This is your actually the target gene, and here primer is. You can snip that portion like that. Okay, and you can say where your situation is, where your gene is situated, and that gene is situated in the TAWGES. So you can click here, so you can found the details, your primer sequence, and also regional sequence, and getting all the information. Okay, and also the sequence ID, which is the gene ID actually. And if you want to get the full sequence of that gene, you can copy that sequence ID and blast in phytosome or NCBI. Okay, like that you can found where your gene is situated.
Okay, sir. Sir, from RNA to cDNA is converted by kit.
Yes,
Okay,
kit
okay.
there is also available, chemical also available, but the main thing is when you are converting from RNA to cDNA, you need to use the same chemical from the same companies. So that's why it's better to use the kit, because all times you are purchasing the different types of chemical in same company. not possible. Okay, so that's why you are can using the kit. It will gives you the best result, and it's also cost effective.
Okay, sir. Thank you, sir.
Thank you. So, any others have any questions? Any participants?
Sir, can you please repeat that plant care site?
Plant care. I think your internet connection is not working too good today,
Yes.
because sometimes I saw you are left the program, maybe due to the internet connection. So in plant care, you just type in Google plant care database. Okay, you found BioTools from plant care. Just click on the BioTools. Okay, sorry. I just forget another thing. Plant care database. I go to the bioinformatics and evolutionary genes, and you can found plant care website. Then search for care. Put your email ID here. If you want to put the ID or anything, you can write here and paste the raw sequence. Here we are putting that sequence, only sequence, no FASTA. So here you just no space or no arrow sign, only the FASTA sequence, and then you can go for search option. You need to wait for 5 to 10 minutes. In your email you can get the result like a zip file like that. After that you need to extract, and within that zip file you found this type of HTML, and within the HTML you can found all the transcription factor. If you want to details of that transcription factor, just click on plus sign. And there also you can found write the function of that. If the function is not suitable for you, you can search in Google.
Okay, sir.
This is the plant care. Okay. So students and participants, if you have any questions any times, you can ask in the group. Okay. If possible to me, I will give you the answer, or other scientists maybe give your answer. If you want to know more about genomic sequence, I also taken all another 7 to 8 classes for genomic sequences, database, and phylogenetic tree analysis, DNA barcoding, transcriptome analysis, GBS analysis. So I'll take step by step. But beside this, if you have any question, or if you want to learn any software, like that one guys asked me about the prokaryotic promoter analysis. So if possible, I'll give you or I'll show you how we can analyze the promoter from prokaryotic. Okay. So like this, if you have any questions, you can write, or if you want to learn any other software or any others analysis, you can also uh, uh, write in group. Okay, I will provide or I will show you. Okay, so thank you today. I think this class may be help to you. So thank you all of you.
Thank
Good
you,
night.
sir.
Thank you.
Thank you, sir, for your valuable classes. And for the participants, the next session will start tomorrow on 6 o'clock. We will share the link tomorrow. And thank you all. Today we are closing the session.